Welcome to week 3, microbiology. This week it's all about clinical virology. So we just have one big PowerPoint here. It's a very large one, I know, all on viruses. So we are just going to jumpstart right away. So let me pull this up. Okay, so in general, we first need to know the characteristics of a virus. So a virus is going to be either classified as RNA or DNA. It just depends on which nuclear material it'll have. It will have a capsid, which is basically the protein coat that encloses that genetic information. It can be different shapes. It can be considered a helical capsule, an icosahedral, or complex. The nucleocapsid is a term just meaning the genome plus the protein coat all put together. Virion is referring to the entire virus. And then finally, envelope. Sometimes a virus will have an envelope and that will surround the capsid. Other times it doesn't have an envelope. So not every virus has an envelope. So the steps to becoming infected with a virus is first, of course, is attachment, penetration, going into that cell, because again, viruses absolutely need cells to exist. They are non-living organisms, although there has been some thought to that that they might not be what we consider dead. Originally, we always said non-living. There is some research right now going on that maybe they're living a little bit, but at any rate, what you need to know is that they have to have a cell. So they are going to penetrate into that cell membrane and uncoat. So basically, they're going to release out that genome, whether it's the RNA or the DNA. And that's going to go into where you would normally find it in a cell. RNA is more found in the cytoplasm. DNA is more in the nucleus. Then it's going to make copies of itself because, it wants to do its thing, survive, do whatever it can just like we do. So it's going to make copies of its genome, produce proteins, so this whole replication process. And it's going to assemble. It's going to start to put it all together into virions, into a complete virus look, and then it's going to release it out. So it's going to let it out, and then it's going to repeat that whole thing again. That new virus is going to go find another cell of its own, attach, penetrate, do the whole thing. So this whole process just repeats over and over and over again. So specimen collection and transport, when are viruses most likely to be present for possible collecting of samples so we can test for it? Well, it's either right before the symptoms or right at the very initial onset. The sensitivity to viral load is kind of rapidly decreasing, so you want to get it right away as soon as possible. Ideal specimens, you can take an aspirated secretion. Swabs, it depends on which virus you're looking for, but swabs can be used. Not the calcium alginate swabs, though. They inhibit the replication and can interfere with certain testing, so that's just kind of a note there. Again, whatever the symptoms that the person is portraying is where you're going to collect the specimen. This is the same when we talked bacteria. If somebody came in complaining of difficult breathing, they were wheezing, they had sputum production, you would get a respiratory sample. Same with this. Same symptoms, let's try to get a respiratory sample. Stool, urine, blood, biopsy, cerebrospinal fluid. So wherever they're complaining, that's where you get the sample. So as much as you can, try to keep the sample or the swab moist because desiccation or drying out can destroy viruses. And then you're using ideally viral transport media. So samples should be cultured immediately. And here are the different types of viruses or antigen viruses that you would look for in each different body system. So, and then a culture type at the very end here that you would collect with those samples. So it's just kind of FYI. It's a nice collection, a nice summary put all together if you like that out of your textbook. All right, as far as testing, there are 3 major methods to testing for viruses. First is directly detecting that virus in a sample, isolating it through cell culture, and serologic assays, meaning blood tests to look for antibodies that you have made to that virus. So that's a pretty common one, especially to certain viruses that we have out there. When you think of hepatitis, we do a lot of serologic blood testing to look for different antibodies that we make to hepatitis. Same with HIV, things like that. All right, so for the direct detection, which is the first one listed here, directly detecting, you can do this through a kit that uses, like, say, an enzyme immunoassay methodology, a direct fluorescent antibody staining, a cytopathic effect from a cell scraping with a microscope. A lot of times our influenza is run through a kit. That's what's showing in the picture on the right. Influenza is a big one to run through a kit. It's just a swab that you go up the nose with, come back to lab, throw it in the kit, follow directions. You look for a positive or a negative on that kit to know if you have influenza A or influenza B. They have cards for both. RSV, respiratory syncytial virus, is done often on a kit. Rotavirus, which causes extreme diarrhea, is done on a kit. So there's lots of kit tests out there. Mono's a kit. So if we can run a kit, so much easier. It's so much simpler. Cell culture, though, is not so simple, and not many places do this anymore. It's very few and far between. But you're basically growing cells to isolate that virus and help detect what it is. Because, again, viruses have to be in cells, so by looking for the cells that have the viruses in there is what you're trying to do. So there are 3 types to cell culture, and you do need to know all 3 of these. First, you have primary cells. So it's basically a lot of times they use what we call primary monkey kidney cells. You're not using very many cell division. It's very minimal cell division occurring, just only a few times before you're going to need to get other new cells to use again. Diploid cell lines can divide up to possibly 50 generations. Typically, this would be a human neonatal lung cell they use here. And then the heteroploid can have continuous division, more than 70 passages. So that's just kind of information on how much the cells really can be used and replicated until we can isolate that virus. And then the serologic assays, again, we are looking for antibodies to the virus. We're typically looking for IgM and or IgG-based antibodies. So going back to immunology, IgM means they actively have the infection if that's positive. IgG means they are having it and getting kind of toward the ending of it, or they're fully recovered. It just depends on what's coming up pause. So this is kind of measuring our host response because we make those antibodies to the virus. All right, so this just determines IgM level equals acute. So acute and convalescent samples, it's saying. Typically, we collect them like 3 weeks apart, I would say. So acute right away at the initial infection, and then 3 weeks later, the convalescent. All right, so now let's actually get into knowing about our viruses. Again, we classify them if they're DNA or RNA-based, and you do need to remember that. So right now, we're going to go through all the double-stranded DNA viruses. So first group is Adenoviridae. These are double-stranded DNA, like I said before. They oftentimes affect the respiratory tract, so pneumonia, eye infections, and GI tract for gastroenteritis. There's not much else I want you to remember on that one. That's a very basic one. Herpes, oh my gosh, huge category. There are so many that fall under the herpes category. You have your herpes simplex virus type 1 and type 2. Varicella zoster, Epstein-Barr, cytomegalovirus, and then you have herpes virus 6 through 8 down at the bottom. So lots under here. So in the herpes virus, again, there is types 1 and 2. Type 1 tends to be waist up, so that's more the oral herpes when you see somebody have them on the lips. Type 2 is below the waist, so that is the STD that we often think of with herpes. There is neonatal herpes, so if the mom were to have it and pass it to the baby upon birth, you get the idea. And then there's herpetic encephalitis, which is when it happens to go up into the brain and cause swelling. So ocular herpes does and is capable of causing blindness. So just a note there as well. I don't expect you just to know everything about the herpes. We're just trying to keep it to the very, very basics here, because there's a lot of viruses we have to cover. Cytomegalovirus, CMV, maybe you've learned this in immunology if you've taken that. If not, if you're with me right now, you're going to learn it. But cytomegalo is passed through different secretions like saliva, tears, urine, breast milk. It can be passed congenitally to the baby, and that's where it becomes the big issues. Anytime it hits a congenital infection or it hits immunocompromised. Otherwise, to healthy people, adults, cytomegalo often feels just like a little bit like a flu. It's kind of like flu-like. You feel tired, you feel run down, you might have a little bit of fever, but it's not too bad for us. A lot of times it's asymptomatic even, but when it hits congenital and hits immunocompromised, it's very dangerous to them, and that's where it causes the majority of problems. We do screen blood products for cytomegalovirus because it is so easily transmitted through blood as well. So oftentimes when people donate blood, that's going to get screened for cytomegalovirus. Epstein-Barr, of course, we know that is the cause of infectious mono, but it's also been linked with causing Burkitt lymphoma and nasopharyngeal carcinoma. We know it's passed through saliva because, mono is nicknamed the kissing disease. There is this one we have a kit for, so we don't tend to culture this. We tend to just screen for it in kit. Or you can do the antibody antigen testing through blood work. So that's the best way to look for the Epstein-Barr virus. So this is just kind of going through when markers are made to it so that we can kind of put it together to know if they actively had it, if they had it in the past, that kind of thing. And that's just showing more on it. I don't expect you to know all of this right now. It's just informational here. Varicella zoster, of course, varicella is chickenpox. Zoster is shingles. They now have a varicella vaccination. So when children are born, like my daughter has the varicella vaccination. Lucky her. I actually had chickenpox, and I think anybody that's for sure my age has had chickenpox. So now when you go to clinicals, they require you to show that you've either had the varicella vaccination or you have to prove it by a varicella titer. So you go get your antibody work drawn, show that you are immune to varicella. If you are not, you have to get the varicella vaccination. Once upon a time when I was in the hospital, we just had to write a little statement. I had chickenpox when I was 9 years old and sign it, and that was our proof. Now they want actual proof. So this is just kind of going through the different about the chickenpox. So it talks a little bit about the varicella, chickenpox with the lesions. And then a secondary reactivation is shingles. Shingle is, of course, always follows a nerve line. It can be very painful. So that's just it. So you don't have to know all of that. It's just informational there. Herpes virus 6 is often called 6 disease or roseola infantum. I cannot say that. So typically just young children resulting in a rash. Then we have Kaposi sarcoma herpes. This a lot of times goes with the HIV patients. Anytime Kaposi sarcoma is heard of, I would bet anything that patient is an HIV positive patient. It just is so rare for anybody else to really get it. And that is it with the herpes. Again, you don't need to know all about all of those diseases ever. Just it's a reminder they all fit in that category and a tish on each one of them. Papilloma category is the human papilloma virus, and this is what we call that causes warts. So genital warts, common warts. It can also cause cervical cancer. So there is now a vaccination available, and my daughter, again, got this. I never got it because I was already past the age when they finally came out with it. So ideally, it's for both males and females ages 9 to 26 years old, and it helps prevent cervical cancer. So if your child is between those ages, they highly recommend that you get it. Otherwise, it's passed sexually. The pox category is smallpox. It's the variola virus that causes smallpox. There are other types of poxes, monkeypox, cowpox, but smallpox, of course, is still thought of as a bioterrorism agent. Even though it's been eradicated, it still could be popping up for bioterrorism. At the time that I wrote this PowerPoint, only the U.S. and Russia had stocks of smallpox. I don't know if that's true or not anymore. I mean, I don't know if anybody's going to really raise their hand and go, we got smallpox too over here, but just an interesting note there. So there is what the smallpox looks like on the skin, highly infectious, which is why they love it as a bioterrorism weapon because it is so easily infectious to the general public. So lesions, those crustal lesions kind of are what gives transmission of the virus. So they used to vaccinate everybody. Now they don't anymore since it's been eradicated, but I believe we have vaccines in stock still. All right, moving on to the single-stranded DNA viruses. We have parvoviride. So this disease, otherwise known as parvovirus, has that slap cheek pattern that's being shown in the picture. So otherwise, a lot of the same type of infection symptoms there. that was it under that one. So that was all the DNA. We're going to move on to RNA, so double-stranded RNA. We have rotavirus. This is a nasty, nasty bugger. Oh, my gosh. So this affects typically younger children. It does say here it can affect elderly as well. So anybody that's a little bit more immunocompromised. This is the most nasty diarrhea you'll see. My daughter had rotavirus. She got it through daycare. It's easily, God, everything gets passed through daycare. She was under a year old when she got it, and she legit had the most liquid water diarrhea for like 9 days. I cannot believe, I can't even tell you how it smelled. It was unreal. Diapers did not keep that in. Nothing kept that in. It was so liquid, it just ran through everything. I mean, it was a great time in our house, let me tell you. So rotavirus, very infectious, lots of daycare passing on that one, but huge diarrhea with it. And you just have to be, I mean, it goes away on its own. It's a virus, it'll take its course. The only thing with, especially with children, like she was under a year, we actually did end up putting her in the hospital for a day just to get her hydration levels up. Because you're passing so much diarrhea, you have to watch for dehydration. And she was refusing to drink anything, eat anything because she felt like crap. And so you have to be really careful on the dehydration part. So she was just put in for IV overnight just to make sure her hydration level stayed good. So that's it. Otherwise, it will just kind of take its toll and go through. It's a good one. I laugh every time I see that. But there is a kit available. My lab had that kit, so they did a quick test on it. Yep, there it was. It was a rotavirus. So, all right, single-stranded RNA. So that was the only one under double-stranded. The rest are single-stranded RNA. We have arenoviridae, which is going to consist of the Lassa virus. That's the only one here I care about. I don't care about the other ones that are listed. So that's it on that one, I think. Yeah, sorry. Bunyaviridae houses the arboviruses, the phlebivirus, which is also caused Rift Valley, and the bunyavirus. I don't get too worried about these ones. This category I don't care that much about, to be honest. We also have the Congo-Crimean hemorrhagic virus. That is a big one that falls under the Bunyaviridae. It's causing hemorrhagic fever. So hemorrhagic fevers are, think of like Ebola. That's one that most of us are aware of, especially with a couple years ago, the whole Ebola scare. But it causes literally bleeding out. That's why they're called hemorrhagic viruses. So this is just another type of one. They're always pretty much high mortality. They usually don't have a cure for it, so they're always really scary. All right, Calciviridae, Norwalk. Norwalk is one that you should know about, especially as healthcare workers or future healthcare workers. Norwalk is a gastroenteritis, definitely spread pretty easily. Anybody that, when this is starting to outbreak and go around, there kind of is little outbreaks of it. If you have any sort of symptoms, you're usually told to stay far away from the hospital, don't go to work, just because it is so much easier to spread. And if you gastroenteritis, you know, think diarrhea. That's what you're getting here. So it talks about how it's spread through in schools, colleges, camps, cruise ships, nursing homes, anything that's closer contact like that. So that's why, as a lab worker, if you have the symptoms of Norwalk and they have a known outbreak right now, they say don't even come to work because they don't want to keep passing it. And I know one of my friends had Norwalk, actually, and she had to stay home, and she was a lab worker. So, all right, so it's just fecal oral, like everything else that causes diarrhea, fecal oral, contaminated food, drinking water, swimming areas, all of the above. There is on this one a kit for, especially during outbreaks, a quick kit that you can get a results on is the easiest way to do it. Coronaviridae is the coronavirus. Coronavirus causes pretty much like respiratory colds, if you think of it that way. So the big one that was under this was the SARS. And I don't know if, I can't remember how many years ago that was that SARS outbreak with Asia. It was such a big thing. So that could be under here. Filoviridae is where your Ebola virus resides, and the Marburg. Both of these are hemorrhagic viruses. So this just talks about Marburg virus a little bit. Marburg was named because it was first discovered in Marburg, Germany, with infected monkeys. But again, high mortality rates, hard to treat. They pass quickly. They cause a panic. You get the idea. Ebola, here's Ebola. So the other ones that fall under this that was filoviridae, excuse me, and then flaviviridae is the dengue. I never know if I say that right. Fever virus, so it's also known as breakbone virus. So severe bone pain is kind of why it's called breakbone virus, just so you know. And then yellow fever virus is also under this one, and St. Louis encephalitis. So you don't have to know much about these last three here. Just know they're under the Flavi. The one that you should know about is West Nile, just because we see West Nile a lot in the United States. So that also falls under Flavi. West Nile is passed by mosquito, causes encephalitis or meningitis, or it could cause a combination of the two, meningoencephalitis. So basically fever, headache, stiff neck, swelling of the brain, that kind of thing. So it is very serious. All right, orthomyxoviridae are the influenzas. So we all kind of know a little bit about influenza. And we have vaccinations to influenza every year. So as healthcare workers, you'll have to get that unless you're like allergic to it or something. But influenza A is probably the one that's caused the most pandemics out of the bunch. So they're listed there. You don't have to know these, but they're listed for you. And then you also have influenza B and influenza C. All right, so I'm just going to skip through those. I can't say this word, Paramyxoviridae. There has parainfluenza viruses, so those are going to be other respiratory viruses as well. So just keep that in mind, but that's all you have to really know. Parainfluenza does cause croup, or the fancy name is laryngotracheobronchitis. So again, croup is that one with that harsh barking cough. There's not much you can do to treat it because it is a virus. You have to just really watch their breathing on children. There's like a respiratory tent they can put them in to help with their breathing. So just know parainfluenza causing croup. So it kind of goes through some of the other respiratory infections here that it causes. I'm not going to get too wrapped up in it. Mumps, of course, we have a vaccination too, but it does cause huge swollen glands in the neck. And then it's very easily transmitted from person to person. All right, measles, also known as rubeola, also vaccination for this. This was a rash all over the body. The big thing was the congenital infection with this one is harsh on the kids. And then RSV, which is respiratory syncytial virus. And this is one that affects children under the age of 2. My daughter, lucky for her, got this one as well. That daycare just gifted her with a lot. Hers was really lightweight RSV though. Again, there's a kit in the lab. Quick came back as a yes. We just couldn't have her in daycare. I was going to say the nursing home, but you couldn't have her in daycare. Hers is pretty light. She didn't have it too bad. There are some kids, again, respiratory. You just really keep your eye on their breathing. The coronaviruses consist of a lot of the enteroviruses. So think of like your gastroenteritis kind of stuff listed here. So poliovirus is under here as well. Coxsackie A, which is hand, foot, and mouth disease. I know one of my friends, her and her son both had that, and she said it was the nastiest sore throat she's ever had in her life. She couldn't believe how bad it hurts. I've heard it hurts really bad. But yeah, that's Coxsackie A, Coxsackie B, I'm not going to worry about. Rhinoviruses, those are the majority of your colds. That's all we're going to say there. Retrovirus, this is where your HIV, and there are 2 types of HIV, type 1, type 2, that both fit in here. And then there's also human T lymphotropic virus that fits in here that is thought to be associated with different leukemias and lymphomas. So a bunch of nasty stuff here. So for HIV, commonly there is a quick kit to screen for this in the lab, and then they can do blood work to look for different antibodies that you can make to these antigens that are listed here. So this HIV virus has the following antigens. It has a core P18, P24, P55 antigens, polymerase enzyme P31, 5166, and the envelope antigens glycoprotein 41, 120, and 160. So there's kind of just a little drawing of where they like to occur. I don't expect you to know this drawing. It's just informational. And there's where they're located again, if they're written a little bit nicer for you on each virus. So glycoprotein 120 is what actually will bind to the membrane of the TCD4 helper cell, and that will enter in. Again, it's a reverse RNA, so you have your reverse transcriptase converting the RNA into DNA. DNA goes into the genome of the cell, and they make new little virus babies. So HIV-1 is the main guy that's responsible for most of AIDS, but HIV-2 exists mostly in West Africa. And they think that there's a lot more than possibly than just these two HIVs. So it is the #1 cause of death in sub-Saharan Africa, 4th leading cause of death worldwide. So it is a big thing to know and think of. So transmission, blood, body fluid, sexual transmission. It is not transmitted by sweat, saliva, tears, urine, milk. People used to, I hope everybody's better now, freak out and think that, oh my God, I can't sit on the toilet seat, I'm going to get HIV. That's not true. Not true. So it was just a misconception when it first came out. All right, so it kind of goes through the disease. I'm not going to read this. You can read it. It's informational for you. I test more on this in immunology than anything else. There's an entire chapter that we'll have on this. Again, you guys already know this from me mentioning it so often. There's a lot of different organisms, fungus, parasites that are well known linked to causing problems with HIV patients since they're so immunosuppressed. Here they're listed again: that pneumocystis crinii, cytomegalovirus, Mycobacterium avium, Cryptococcus neoformans, Toxoplasma, tuberculosis, that Kaposi sarcoma, and their list just goes on and on. But these are some big ones that are known to cause infections. So here's a bigger list for you, if you will, just informational. All right, to diagnose, Western blot is our confirmatory test. So it is actually looking for those antigen markers there. Again, you can do enzyme immunoassay kits to quick test and screen. You have to confirm it. All right, so that's it for those. We'll move on. Rhabdoviridae contains a listvirus, which is responsible for rabies. So your animal should have the rabies vaccination. So just kind of goes through that. Lots of CNS involvement in the of rabies, paralysis, excessive salivation. That's the whole thing with foaming at the mouth. They're scared of water, blah, blah, blah. So you don't have to know a lot about it. You just need to know the listavirus under Rhabdoviridae is rabies. Rabies and rhabdo to me look similar, so that's how I always remember them. Togaviridae consists of the rubella virus. I don't care about the other ones. Rubella is your German measles. So again, a nice rash all over. Some arthritis that could be seen. Big one with congenital disease. It's very harmful to the baby. And your hepatitis viruses. There is 6 of these little suckers. They're not so little, I guess. 6 of them, but here's kind of just in FYI on a little few of them. The only one here that's listed here is hepatitis A, that's foodborne. All the rest here are more blood body fluids on this chart. So fecal oral route for HIV causes diarrhea. It does have a vaccination available, so if you're going to a country that is more prone to hepatitis A, you would get the vaccination first. Hepatitis B is a DNA virus. The rest of the hepatitises are RNA. That's why they're under the RNA category. But take note that hepatitis B is a DNA-based virus. So this, of course, we have a vaccination for, thankfully, now. And as healthcare workers, it's highly recommended that you get the vaccination. We can't mandate it, but I would highly recommend it. That's the one that takes 3 vaccinations over 7 months to get completion of. Again, hepatitis B hugely transmitted through blood, blood products, sexual activity, and so on. So there's all the risk factors of people, healthcare personnel listed right there. All right, so here is a summary of the markers, and we're going to learn this in immunology. So if you have not taken immunology yet, we will get it there as well, but you should be aware and familiar with these Hep B markers. So anytime that somebody first gets hepatitis B, they will make the hepatitis B surface antigen. That is the very first clue that somebody actively has the infection. Hepatitis B-E antigen means that you're actively replicating, you're highly infectious. The antibody to hepatitis B core is the very first antibody made. If you have the IgM antibody, you still have the active infection. If you only have the IgG antibody to the core, you're starting to recover. The antibody to hepatitis B-E means you're, again, recovering. And then finally, if you have been vaccinated, your blood work should show you have the antibody to hepatitis B surface. That indicates you've either fully recovered from the disease or you are immune because you've had the vaccination. So know each one of these markers and what they mean. So they're listed out, but I would memorize that list there. All right, hepatitis D, which is the delta hepatitis, you have to have hepatitis B in order to get hepatitis D. It's a co-infection. The delta hepatitis requires hep B so that it can replicate and do its job. So you can't have it on its own. It goes with hep B, co-infection. And hepatitis C has becoming much bigger in prevalence. This is one that I hope at some point they make a vaccination for because it's becoming more and more commonly seen. Again, blood and blood products, sexual contact. And again, we can look for the antibodies, the IgM antibody, the IgG antibody to see if you have active or you're recovering from an infection. There is a screening kit, and then again, you're always confirming. Hepatitis E is similar to hepatitis A with the fecal oral content, drinking water, contaminated, that kind of thing. So out of all the hepatitis, A and E are the only ones that are fecal oral routes. All the rest are blood and blood products. And then finally, prions. I know a prion is not a virus. It is considered a protein infectious particle that causes a group of diseases that are these encephalopathies. So they cause encephalitis. Creutzfeldt-Jakob is probably the big one out of here coming from a prion. Just do note what a prion is, Creutzfeldt-Jakob as an example of it. Definitely very harmful, very hard to get rid of prions. Leads to brain damage, rapidly progressive, and it's typically fatal. Otherwise, other forms of prions, what they cause bovine spongiform encephalopathy. So this is kind of like a variant Creutzfeldt-Jakob when you eat infected meats. Otherwise, there's animal ones as well. So there's the symptoms. It's a lot of CNS, central nervous system type symptoms because it goes to the brain damage and things like that. So dementia, memory loss, behavioral issues, tremors. So anything your nervous system controls with your muscles, your speech, your memory, all that is kind of going to be going. Transmission is through neural tissue. So if you eat something that's infected, things like that. All right. Oh, my goodness. So I didn't know I had review questions here. Oh, look at that. I'm not going to go through these. I'll let you guys go through these on your own because I feel like you've been listening to me forever. If you have a question on these, let me know. If you're in immunology, please wait on these because we're going to answer them together and when we get through this chapter. But you can look at them and answer them to help you review. So otherwise, if you're in immunology, we're going to get to them eventually at that point. Okay, so for the virology chapter, so much to know. Know the examples that fall into each class. Know if it's a DNA or RNA-based virus to begin with. If I mentioned a disease, you don't have to know a ton about the disease. That's not what we're trying to get at here. Just know which ones fit into which categories. The information at the beginning before we started getting there is important. You should know these characteristics of viruses on this slide. You should know its steps of infection. That's kind of it on those few. This is kind of informational. And then you definitely need to know the 3 major testing methods and the cell lines here for the cell culture. So do you know that along with the viruses and their categories and which disease fit into which category? All right, best of luck to you guys. Thanks so much and have a great week.